all, welcome to Physio Exam Buddy. This video is on cell-mediated immunity. The cell-mediated immunity is mediated through activated T lymphocytes. You know that there are two types of lymphocytes in the body, T lymphocytes as well as B lymphocytes. Out of this, the activated T lymphocytes, they mediate the cell-mediated immunity. And your answer should begin with the four different types of T lymphocytes. So, which are the four different types of T lymphocytes? They include helper T cells, suppressor T cells, cytotoxic T cells as well as memory T cells. And out of this, the first two, that is a helper T cells and suppressor T cell, they together constitute the regulatory cells of the cell-mediated immunity. First, in detail about the different types of the T lymphocyte. First, about helper T cell. Out of the total T lymphocytes, 3/4 it belongs to the group of the helper T cells, and they form the major regulator of all the immune functions, and they also form a series of protein mediators, that is either lymphokines or cytokines. And the examples of lymphokines in our body are basically interleukin 2, 4, 5 as well as 6, and granulocyte, monocyte, colony stimulating factor, and interferon gamma, etc. Out of this, the interleukin 2 is the one which causes the stimulation of the growth and proliferation of cytotoxic T cells as well as suppressor T cell. Whereas the interleukin 4, 5 and 6, it is basically related to the humoral immunity, because these three, it stimulate the growth and differentiation of plasma cells and antibodies, which is related to the humoral immunity. Whereas the interferon gamma, they will activate the macrophages. And there are two subtypes of helper T cells. First one is Th1 cells as well as the second one is T helper 2 cells, that is Th2 cells. The Th1 cells or T helper 1 cells, they secrete the interleukins, that is the lymphokines interleukin 2 as well as interferon gamma, and they are mainly concerned with the cell mediated or cellular immunity. Whereas the subtype of the T helper cell, that is T helper 2 cell, they secrete the interleukin 4 as well as 5, and it is related to the humoral immunity. And one of the peculiarity of the helper T cell is that it displays the glycoprotein marker, that is CD4. CD4 T cells. What is the full form of this CD? It is clusters of differentiation. That is, it is the basis of their reactions to a panel of monoclonal antibodies. So here the helper T cell, they display a glycoprotein marker, that is cluster of differentiation CD4, that is CD4 T cells. Next is about the cytotoxic T cells. They are also known as killer cells because they release cytotoxic as well as digestive enzyme into the attacked cell, and finally it will destroy it. And the cytotoxic T cell, they display the glycoprotein marker CD8, that is CD8 T cells, they are the cytotoxic T cells, whereas CD4 T cells, they are the helper cells, which can be asked as an MCQ question also. And they have receptors on the cytotoxic T cells, which will bind to the antigen on the microorganisms, and these will secrete a certain proteins which is known as perforins, which is again asked as an MCQ. The third type of T cell, that is the suppressor T cell. They usually suppress the function of the cytotoxic T cell as well as the helper T cell. So this helps to prevent the cytotoxic T cells from causing damage to the body's own tissues, and this is responsible for the immune tolerance mechanism. The last type of T cell is the memory T cell, that is on first exposure to a given antigen, some of these T cells, they become the memory cells. So on a second encounter with the same antigen, they will be these memory cells, they are converted it into effector cells, and they produce an accelerated response to the second exposure to the same antigen. So that is about the memory T cells. Now we will discuss the mechanism of the cell mediated immunity. So the mechanism of cell-mediated immunity you have to explain under these headings. The first one is that whenever a microorganism enters into your body, the antigen processing as well as presentation, that is the first step of the cell-mediated immunity. This will be followed by the antigen recognition by our T lymphocytes or T cells. And then the third step is the activation of these T cells, and they form a clonal proliferation as well as differentiation into memory cells and effector cells. And these are the 5 steps in the activation or the mechanism of the cell mediated immunity. And this is the diagram you are supposed to draw along with the cell mediated immunity. We will explain in the coming slides about this diagram. So the first step, that is the antigen processing and presentation. On exposure to an antigen, that is whenever any microorganism or the foreign body enters into your body, the T cells will be proliferated and it is finally released and distributed into our body. And these T cells will act only against those antigens which is bound to a specific glycoprotein, and that glycoprotein, it is known as the MHC protein or human leukocyte antigen protein. MHC means major histocompatibility protein or human leukocyte antigen protein. So our T cells will proliferate and it will get released and distributed on exposure to any foreign body or antigen, and this T cell will act only against those antigens which are bound to a specific glycoprotein, and that glycoprotein is the MHC, that is major histocompatibility protein or the human leukocyte antigen protein. And the genes of this MHC, they are located on the short arm of the human chromosome. So the first step, that is the antigen recognition as well as processing. That is the antigens who bear the particular protein, MHC protein or the HLA protein, will only be recognized by our T cell. So this is the antigen which bear the MHC2 protein. So then only the antigen will be recognized by our CD4 helper cell. And this MHC proteins which are seen on the surface of an antigen presenting cells. So what are these antigen presenting cells? The antigen presenting cells, they usually have the major histocompatibility protein or HLA antigen on its surface. And the most commonly seen antigen presenting cells in our human body are, first one is dendritic cells, which is present in the lymph nodes and the spleen. They belongs to the most potent category of the antigen presenting cell. And other antigen presenting cells in our body are Langerhans dendritic cells in the skin, macrophages as well as B cells. And this MHC protein which is present on the antigen presenting cell, they are of two types. First one is MHC1 and second one is the MHC2. This MHC1 or the major histocompatibility complex 1, they are present in almost all nucleated cells in the body as well as platelets, and they present the intracellular antigens to the cytotoxic T cells. Whereas MHC2, they are present only on the immunologically active cells like our B cells, macrophages as well as other antigen presenting cells. But here the difference is that MHC1 actually presents intracellular antigen, whereas MHC2, they usually presents the extracellular antigens, that is the antigens present outside the cell like bacteria to helper T cells, whereas MHC1 will present these antigens to the cytotoxic T cell, whereas MHC2 will present the antigen to the helper T cells. And the identical things only have the same set of human leukocyte antigens, and they also have a role in the recognition of the tissues as self or foreign. So this is the main factor in the rejection of the transplants. Now, next step is the antigen destruction by our cell-mediated immunity. So, once an antigen bearing this major histocompatibility complex enters into the body, they will be taken up by our antigen-presenting cells. They are only partially digested inside the antigen-presenting cell, and these digested particles, they will be transported to the surface of the antigen-presenting cell. And the digested particles which is present on this surface of the antigen presenting cells, they are then presented to the activated T cells in combination with the MHC, that is the activated T cell bearing the major histocompatibility complex will bind with the digested particles which is transported to the surface of the antigen presenting cells. So the MHC1, that is major histocompatibility complex 1, they usually presents the antigen containing cell to CD8 plus T cells, whereas MHC2 they presents the antigens to the CD4 T helper cells. That is CD8 T cell is the cytotoxic T cell. So MHC1 will present the antigen to the cytotoxic T cell, that is a CD8 T cell, whereas MHC2 will present the antigen to the CD4 T cell, which is the helper T cell. And this CD4 and CD8, these proteins facilitate the binding of the major histocompatibility complex to the T cell receptor. So this antigen presenting cell, I have already told that it bears either MHC1 or the MHC2, and this MHC, that is major histocompatibility complex, will bind to the T cell receptor, that is represented as TCR here, which is present on either the CD4 cell or the CD8 cell. And the T cells that survive the thymic processing only possess these T cell receptors, and these T cell receptors have two properties, that is they have the ability to differentiate between foreign as well as self major histocompatibility complex, and they also react to the complex of antigen and self MHC molecule. In detail about the T cell receptor. It is a multi-subunit complex, and you can see here that it is made up of two chains, that is alpha chain as well as beta chain, which is linked by a disulfide bond. And there is an antigen recognition site, and this T cell receptor are usually surrounded by an adhesion protein, and they usually will bind to the complementary protein on the antigen presenting cell, and they form an immunological synapse. So the synapse between this T cell receptor with the major histocompatibility complex, this is an example for immunological synapse. And for the T cell activation, two signals are required. The first one is that the binding of the digested antigen should happen with the T cell receptor, and there should be a formation of the immunological synapse. If the first signal occurs but the second signal, that is formation of the immunological synapse, doesn't happen, the T cell won't be activated. That is, the T cell will be inactivated and it become unresponsive. So for T cell activation, there should be two prerequisites, that is two of these signals should be required. First one is binding of the digested antigen to the T cell receptor as well as formation of the immunological synapse. And the next step following the antigen destruction is the clonal proliferation as well as differentiation. Usually this clone of cell, that is the combination of antigen presenting cell bearing the major histocompatibility complex, when it binds with the CD4 or CD8 receptor having the T cell receptor. So when these combination of cells, they join together and forms the immunological synapse, there occurs a clonal proliferation of these type of cells, and finally it will differentiate into either Th1 helper cell, Th2 helper cell, which are the two subtypes of the helper cells, or as well as the memory cells. But for cell mediated immunity, you should only know this portion, that is after clonal proliferation, differentiation will occur into the Th1 cell, that is Th1 will probably differentiate into interleukin 2 as well as interferon gamma, whereas the interleukin 1 as well as interleukin 2, they will proliferate into the CD8 cell and T regulator cells, whereas the interferon gamma will activate the macrophages. So this is the step which follows the antigen destruction by the T cell, that is whenever any antigen presenting cell having a major histocompatibility complex binds with the T cell receptor have on the CD8 or CD4 cells, they first undergo clonal proliferation, then finally they will differentiate either into Th1, Th2 and memory cells. But in cell mediated immunity, you require only this portion, that is these clonal proliferation of the cells in cell-mediated immunity, they will differentiate only into Th1 cells, which include interleukin 1 and 2, as well as interferon gamma. Out of this, the interleukin 1 and 2, they will differentiate into CD8 T cells as well as T regulatory cells, whereas the interferon gamma, they will activate the macrophages. Next, about the effector cell function. So we have already seen that the interleukin 1, 2 as well as interferon gamma, they will form the effector cells, that is CD8 T cells, T regulator cells as well as macrophages. Out of this, the CD8 T cells, they secrete the protein perforins, which will help in the lysis of the target cells. Whereas the second one, that is the T regulatory cells, usually regulate the actions of our helper as well as cytotoxic T cell. And the macrophages, it will causes the enhanced killing of the phagocytosed bacteria. So by these three mechanisms, the cell-mediated immunity will kill all the viruses, tumoral cells, funguses as well as intracellular bacteria. So this is in short about the mechanism of the cell-mediated immunity. So this is the diagram you are supposed to draw along with the cell-mediated immunity. That is, first of all, if an antigen enters into your body, it will be recognized as well as it will be processed, and then it will undergo activation. And these cells, this cell, that is the combination of antigen presenting cell containing the major histocompatibility complex, when it binds with the CD4 or CD8 cell bearing the T cell receptor, they undergo clonal proliferation, that is, they will multiply. And in case of cell-mediated immunity, this clonal proliferation will differentiate into Th1, that is helper T cell 1 subtype, and from this it will causes the differentiation of interleukin 1 and 2 into CD8 as well as T regulatory cells, and the interferon gamma will activate the macrophages. Out of this, the CD8 T regulatory cells, T cells, they will secrete the perforins, which causes the lysis of the target cells, whereas the T regulatory cells, they will regulate the actions of the helper as well as cytotoxic T cell, and the interferon gamma, which differentiate or activate the macrophages, they will enhance the killing of the phagocytosed bacteria. So by all these three mechanisms, the cell-mediated immunity will kill all the viruses, tumor cells, fungi as well as intracellular bacteria. Thank you.